Hello friends, how are you doing? And I hope you are doing great and things are fine and absolutely awesome, right? So we are doing the chapter excretion. We are doing the chapter excretion, and let us see, 1 to 2 to 3 minutes. We'll just take a quick recap of what we did last time. We talked about, we said that yes, there are two types of nephrons. First of all, we said what is the structural and functional unit of a kidney. We said nephron, and we said that there are about 1 million nephrons in each kidney, 1 million nephrons in each kidney, and in one kidney, and this nephron is a structural and functional unit of a kidney. Structural and functional unit of a kidney, just just similarly to how the cell is a structural and functional unit of any living organism. Okay, the nephron, also called as a uriniferous tubule, we said has many parts, starting right from the the Bowmans capsule to the proximal convoluted tubule to the loop of Henle, distal convoluted tubule, and the collecting duct. Then we said that yes, we talked about the lumen of the artery of the afferent and the efferent arteriole. Then we talked about the difference between the two types of nephrons, the cortical and juxtamedullary, and we said the cortical nephrons are 80%, and the juxtamedullary, which is found only in birds and mammals, are just 20%. Right. So we have not talked about the Malphite capsule or the renal capsule. We'll just talk about it. So we are taking the Bowmans capsule in a little detail, not that much. These are still not much detail. Don't have the misconception that we're just doing that much so that we can understand. Okay. Right. Right. Why give the space? We will just talk about it. So this we all know is the Bowmans capsule. Bowmans capsule. Right. Yes, the Bowmans capsule. Now we all know that these are what? These are the capillaries. Right. The glomerular capillaries. Now, as I told you, this Bowmans capsule has two layers. One is the outer parietal layer. The outer parietal layer of Bowmans capsule, right? The outer parietal layer Bowmans capsule made up of epithelial squamous epithelium, made of squamous epithelium. The outer parietal layer of Bowmans capsule made up of squamous epithelium, and this one is the inner parietal layer, inner parietal layer of Bowmans capsule having cells called the podocytes, having cells called podocytes. Okay, can you see this whole thing over here? Right. So I'll just draw these, so that it will help you. So rather than drawing this outline, you could have just drawn the cells. Okay, and this here, what will we see here? So these are the podocytes, as I told you. This is the parietal layer. Now, so how are the podocytes? If I draw that one podocyte, somewhat like this. This is a podocyte, and this is what? This is some feet-like structure, right? So these are the podocytes which is making up the inner parietal layer of the Bowmans capsule, and the slits which see in between, these are called as the filtration slits. Filtration slits. Now what happens? Why are these filtration slits present? So that when the filtrate which has been filtrated out comes out from these capillaries, they can get into this, in this, and continue into the proximal convoluted tubule and further into the loop of Henle, right? So this is how the whole thing. Now what we see, so is this what we have? This is not what we have. Here there is a basement membrane. Okay, there is a basement membrane. So this is the basement membrane. Right? I think the structure is looking beautiful. Yes. Now what we see, so this Bowmans capsule with this Bowmans capsule, right? Can you see the whole thing in one frame? This Bowmans capsule with the glomerular capillaries is together referred to as Bowmans capsule plus glomerular capillaries is together referred to as the Malpighian capsule. Malpighian capsule or renal capsule or renal capsule. Right? Visible till here? So the Bowmans capsule plus the glomerular capillaries is called the Malpighian capsule or renal capsule, and what we will see that this, the glomerular capillaries, okay, now I have to, I'm not, I'm not liking to rub this. This is so beautiful. Okay. So what we can say, the glomerular capillaries, the glomerular capillaries consist of about the glomerular capillaries consist of about 50 capillaries, consist of about 50 capillaries. Right. Now let us talk about the filtration membrane. Let us talk about the filtration membrane. So what does the filtration membrane consist of? When you're talking about the filtration membrane, what we will see that, yes, arteriole, bring that picture, the picture which I just dropped into mind. Okay, we said that yes, there will be the podocytes present. Right, and then we said that these are the podocytes, and so there will be then these are the endothelial cells. Now the capillaries have just a single layer of endothelium, and in between these endothelial cells there are again pores here which are called the fenestrae, and here will be the basement membrane. So this whole comprises of the filtration membrane through which the filtration will take place. This whole comprises of the filtration membrane through which the filtration will take place. What are these? This is the endothelial cells, of so these are the endothelial cells of glomerular capillary, right? Endothelial cells of the glomerular capillary, and what we see, these pores, the pores are called as fenestrae. The pores are called as fenestrae. This is the basement membrane these are the podocytes, and as I told you, between the podocytes there will be the filtration slits, filtration pores or slits, whatever you want to say. filtration pores or slits. So come back to this part. This whole thing is a filtration membrane, and this is how you know, and this is from where the filtration will take place. So how will the filtration take place? The filtration is taking place from where? The glomerular capillaries, and coming where? Coming into the renal tubule, right? Renal tubule. So the single layer of cells, you know, present in the capillary, in the glomerular capillary, is called as the endothelial cells. Between the endothelial cells there is leakage, you can say, and these are called as the fenestrae. Yes, those are called as the fenestrae. Now what we see, you know, this is a basement membrane. So and finally what will happen, you know, this will come through this, this will come through this, and these will enter through the filtration pores or slits present between the podocytes. Right, so this whole comprises the filtration membrane. Right, we're not writing, we're mostly doing the you know, the diagrammatic part. Right, what else can we talk about? What else can we talk about? Also we can say that yes, the filtration, so we have talked about, let us now start talking about the proximal convoluted tubule. Proximal convoluted tubule. Now we say that the proximal convoluted tubule have a brush bordered epithelium, okay. These have a brush bordered, okay, these have a brush bordered epithelium, okay. These have a brush bordered epithelium. Why brush bordered? Because, you know, see, these have cuboidal cells with numerous microvilli, okay. So cuboidal cells we are taking, isn't it? So if you talk about a whole column of cells, isn't it looking like a brush, the tufts of the brush? So this is why this is called as the brush bordered epithelium. These have a brush bordered epithelium called as brush bordered as numerous cuboidal cells, okay, as numerous so called as brush bordered as numerous cuboidal cells lined with microvilli, okay, lined with microvilli. The proximal convoluted tubule is in continuity, this tubule is in continuity okay. So this tubule is in continuity with what? Is in continuity with the loop of Henle. Also, cells of proximal convoluted tubule have a lot of mitochondria as the PCT is very much involved, as proximal convoluted tubule is very much involved in active absorption. Okay, so this is the proximal, and so what is absorbed here actively? 100% glucose, 100% amino acids, 70 to 80% of sodium and potassium ions are actively absorbed here. %, glucose, 100% amino acids, 70 to 80% of sodium and potassium are actively absorbed here. Where? In the proximal convoluted tubule. That is why these active absorption needs energy, and what provides energy? It is the mitochondria which will provide you energy. Okay, it is the mitochondria which will provide you energy. Right. So what next? Then we can talk about the descending limb. Okay. The descending limb. Okay, descending limb. Now, when we are talking about the descending limb, what we will see that these are lined with squamous epithelium, right? These are lined with squamous epithelium. These are thinner as compared to the ascending limb. These are thinner as compared to ascending limb. And then thirdly, these will be, okay, these are impermeable, these are permeable to water, these are permeable to water and impermeable to sodium and chlorine, to basically ions, and impermeable okay, to ions like sodium and chlorine, like sodium and chlorine. So these are permeable to water and impermeable to ions like sodium and chlorine. Then we can talk about the ascending limb. these are lined with cuboidal epithelium, lined with cuboidal epithelium, right? Definitely, since this will be thicker, and what we see, impermeable to water, but permeable. So just the opposite of the descending limb. Ascending limb is just the opposite. Okay, here squamous epithelium, here cuboidal epithelium. This is thinner, this is thicker. This is permeable to water, impermeable to water. This is impermeable to iron, this is permeable to ions. Impermeable to water but permeable to ions like sodium and chlorine, right? Then we can talk about the distal convoluted tubule. Distal convoluted tubule. Now the distal convoluted tubule, okay, these have cuboidal epithelium, these have cuboidal epithelium, right? These have cuboidal epithelium, and also what we will see in the distal convoluted tubule, what we will see that these have some microvilli, and are permeable to sodium and potassium, and then coming to the collecting duct, what we will see, lined with columnar epithelium, right? So in this session we have talked about many things, right from the structure of the Bowmans capsule, the Malpighian, what is Bowmans, what is Bowmans capsule, what is the Malpighian capsule or renal capsule, and then we talked about the different this thing, that how what is the function and what are the structure of the nephrons. We talked in detail about each part of it. In the next session we'll talk something more interesting. The time we meet again, a very goodbye from this side. Thank you.